Okay. Okay, so today we're going to kind of start making our transition into the nervous system. Okay, this is really kind of, we're not quite there because Friday we're going to talk about muscle metabolism, but this is kind of a transition day where we talk about really the connection between the nervous system and skeletal muscle, which is known as the neuromuscular junction. And we'll talk a little bit about membrane potentials, which is a big piece of understanding the nervous system. Okay, so the neuromuscular junction, again, it's this place where our nerve or our neuron meets a skeletal muscle. Okay, and it has three separate components to it. Okay, so we've got kind of like the nerve side of it, which is called the presynaptic terminal. Okay, this is the end of a neuron. Okay, a neuron is a cell of the nervous system, right? Okay, those are the ones that have the long axons. The very end of that axon, it's going to kind of expand in the end, and it's going to contain a bunch of neurotransmitters and some other components that allow it to do its functions. And so, with our motor neurons that are innervating our skeletal muscle, they're going to contain a bunch of acetylcholine. Acetylcholine, always abbreviated as just ACH. That's our primary neurotransmitter of skeletal muscle. Okay, so anytime a nerve is activated in skeletal muscle, it does that by releasing acetylcholine, and that acetylcholine binds to the skeletal muscle. Okay, second part of the neuromuscular junction is just the space between the two, between the nerve and the muscle. Okay, that's called the synaptic cleft. This is where that neurotransmitter diffuses through. So it's just a fluid-filled diffusion space. Okay, it's going to contain a certain enzyme called acetylcholine esterase. That's like the name kind of implies, it helps us break down acetylcholine. Okay, and the why of that is because that's going to help muscle relax once the signal from the nervous system has stopped. If acetylcholine just stayed there all the time, then our muscle would just keep contracting. All right, so it has a certain half-life, and that half-life is sped up and made shorter by this enzyme helping to break it down. Okay, and then finally, on the muscle side of the neuromuscular junction, it's called the motor end plate. Okay, so this is a specialized portion of the cell membrane of the muscle cell. Cell membrane is called the sarcolemma, right? So it's just a special portion of that, very folded, and it contains a bunch of receptors for acetylcholine. Okay, one thing we'll see here in a sec is that acetylcholine receptors are also ion channels. So acetylcholine is going to go bind to that protein. It's got a perfect little spot for acetylcholine to fit onto it. When it binds to it, it opens it up, and then ions are able to pass through. And that's how we kind of send the signal from the nerve to the muscle. Okay, so take a sec with the people on your team and label these different structures in the neuromuscular junction. And then you're going to send in the letter for the place that acetylcholine binds to the muscle cell. Okay, so we're going for the letter there. 1, 2, 3. Those are the just kind of the regions. And then you've got the other structures there in the letters.
Oh, wait, no, one is the piece. Sorry, one. There's one on here, but is A vegetables?
Yeah, that's called acetylcholine. Yeah. Yeah. All right, so you're sending in your answer there to the question on top of the slide. Where does acetylcholine bind to the muscle cell? Just as a reminder too, I would love to see cell phones in your bags, not on your desk. I know how tempting those little guys can be. They're just sitting there talking to you, wanting you to flip it over and look at it. Thanks. Helps you stay engaged. Just trying to help you out. Okay, what do we got? 58, anybody else? Let's get a few more people there, I think. Good. All right, I got 66 now. 67, we're almost there. All right, that might be about everybody. Got 68. Okay, so the vast majority of you are saying C for that answer. Okay, so C is pointing to, yeah, so it's pointing to that kind of pinkish receptor there, right? So that is our, bless you, that is our acetylcholine receptor for sure. Okay, so good. Okay, we got 69 people. I'll leave it open for just a sec. All right, so let's start with the letters first. So A, what do you guys think A was? Yeah, so it's a vesicle full of acetylcholine. Okay, so yeah, so we got that. And then B, any guesses there? It's just a little blue dot. So how would you know, right? It's acetylcholine esterase is what it's being depicted there. So that would be that enzyme that helps us break down the acetylcholine. C, you already said, was the acetylcholine receptor. This is a version of an ion channel, right? It's called a neurotransmitter gated ion channel, right? So it's a channel that's going to allow for ions, positively or negatively charged ions, to flow through it, but it's gated by something binding to it, right? The key to open it up or close it is something binding to it. Okay, so in this case, it's a neurotransmitter. Also, this is also known as a ligand gated ion channel. Something binds to it, opens it up. Okay. All right. And then we got, what else we got? We got D. D is a different type of ion channel that we're going to talk about coming up here pretty soon, called a voltage gated sodium channel. So another version of an ion channel is going to let ions pass through, but the thing that changes its state or opens or closes it is a change in voltage in the membrane, not something binding to it. So when something binds to it and changes its state, that's called a ligand gated ion channel. If it's the change in electrical kind of current around it that changes it, then it's called a voltage gated ion channel. Okay, and then we've got E over there. That's another version of a voltage gated channel. That is a calcium channel. Okay, we'll talk about how it's involved in this process here in just a sec. But again, when the voltage around that channel changes, it opens up and then calcium is able to flow into the neuron there. Okay, and then lastly, F, what do you think F was? Acetylcholine, right? That's our neurotransmitter, okay? It was in those little vesicles. Those vesicles kind of migrated towards the surface, fused with the membrane, and then just dumped their contents out into that synaptic cleft. Okay, so then the 1, 2, and 3 are just those regions of the neuromuscular junction we talked about. What's number 1? What do we call it? Presynaptic terminal. There we go. Okay, and then number 2 is what? The cleft, right? Synaptic cleft. And then number 3, motor end plate, right? So that's a specialized part of the muscle cell membrane. Okay, so this is just kind of the anatomy of the neuromuscular junction. Again, this is the nerve side up here, right? This is the muscle down here, right? And then this is just the space in between the two. Okay, questions about any of those structures or any of this stuff so far? Okay, all right. Okay, so let's talk a little bit about voltage across a membrane, because this is a very, very key part of the nervous system. The nervous system is really ran by changing voltages across the membrane. Okay, signals that our brain sends to our muscles or to any other structure. It's all about changing voltage across a membrane, which is really just like the charge difference from one side of the membrane to the other. Okay, and so in order to have the ability to change the charge, we have to have a starting place in all of our cells, and that's called resting membrane potential. Okay, so where is the charge situation typically at when a neuron or a muscle cell is at rest? That's called resting membrane potential. Okay, so we can see this is our cell membrane here. Okay, on the right is inside the cell, right? That's intracellular. On the left is extracellular. That's outside the cell. Okay, we can see right here that there's a lot more potassium on the inside than there is on the outside, right? You guys see that? See the purple little potassiums? There's way more of those on the inside, just a couple on the outside. And it's just the flips of that for sodium, right? There's a lot more sodium on the outside of the cell, not much on the inside, okay? This is a key thing that helps us do all of the cell signaling that we do with the nervous system, okay? So inside's got high potassium, and outside's got high sodium. Okay, the inside also has these things, these A's with negative charges. Those are trying to represent negatively charged proteins. Okay. Yeah, and we got low sodium on the inside, low potassium on the outside. Okay, so that's kind of our starting place. So then what this ends up doing is the inside, as it shows right here, is more negative than the outside. Okay, so that's one of the key things from this slide, is that the inside of our nerves and our nerve cells and our muscle cells is more negative than the outside. That's part of resting membrane potential. Okay, and then the other key thing here is knowing that the primary ion of our intracellular space is potassium. The primary ion of our extracellular space is sodium. Okay, got those three things? Okay, all right. So then, yeah, so we've got it here. Okay, let's just do a quick check of that to see, make sure we're on the same page. Okay, this is showing another situation here that helps to maintain this resting membrane potential. But just check with everybody on your team and send in the answer to that question on the bottom. So resting membrane potential is -70. Okay, what does that actually mean though? Does that mean the inside's negative or the outside's negative? Okay, so talk with the people on your team, make sure you're on the same page, and then send in that answer when you're ready. Okay, I got about 10 more people need to send it in, it looks like, about 10 more. All right. I think that's everybody. So good. So almost all of you, 96% said A, that the inside is more negative than the outside. And that is absolutely true. Okay. The image on this slide is showing you one of the key things that keeps it that way. Okay, this thing is called the sodium potassium pump. Okay, it's just this little protein we find on all of our electrically excitable cells, all of our neurons, muscle cells. What it's constantly doing is pumping 3 sodiums out and two potassiums in. Okay, it uses ATP to do this. Okay, so it's pushing these ions against their gradient. Okay, and so what type of movement is that? What do we call that? When we're pushing something against its gradient, what do we call that? Active transport, but then specifically if we're using ATP, which type? Primary, yeah. So primary is when we're using ATP. Secondary is when we're using the gradient of something else, right? So this is primary active transport. this accounts for just this guy right here accounts for at least around half of the energy usage of our nervous system. Just pumping sodium and potassium back and forth. Okay, so it obviously has a key role to play. Okay, I'm gonna see if I can get this video to work on my computer here real quick. There's a nice little description of resting membrane potential. This typically doesn't work on my iPad, but maybe we got a chance today. Nope. Give it 1 sec. I don't know why the iPad's so bad for that these days, but let's switch over to my laptop, and I think that'll work. Okay, now I remember the volume has also been bad in this room, so let's see if we can hear it.
The human brain alone contains about 100 billion
Let me try that one more time. I'll just do it from my computer if we have to, but speakers should really work in this room. That's not what I meant to do. Yeah, we'll just go from my laptop then, like we did last time.
The human brain
All right,
alone
here we go.
contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium lumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
All right. So let's go ahead and just go right to that clicker question on the next slide and discuss that with your team. And you can send in the answers for that. It's going to be a choose all that apply type question. So discuss that and send in the answers that you think are correct.
potentials are a change in the charge.
The idea of having one
Yeah. I feel like since it makes more sense for the more. And then outside, that makes sense for the ending to
So the normal membrane potential is -70, so when it's at 0, that means there's more potassium?
Zero, what do you mean zero?
Like when the voltage measures 0, what does that signal?
That would mean that it's moving in the positive direction, right? And we haven't really talked about changing it yet, but
Okay,
we'll get there. But,
okay.
yeah.
I wasn't sure, 'cause I know in the video they were talking
Mhm.
about when
Oh.
it measures 0.
Oh, yeah, yeah, don't worry about that too
Okay.
much yet, but yeah, we'll get there. We'll talk about changing and depolarization a little bit later. Yeah. Okay, so go ahead and send in your answers. If you haven't yet, you can send in more than one. All right, make sure you hit send on this type of question. I still need about 10-ish more of you, 12 more. Haven't sent it in yet. All right, so we've got 84% of you saying C. Okay, so that's one that a lot of you are choosing. 75% of you saying A, and we've also got about half of you saying E. Okay, so let's talk through these a little bit. So this one A says the fact that there's more passive leakage of potassium than there is of sodium. Okay, so if we've got a bunch of potassium on the inside, right? That's the case, not much on the outside. If it's allowed to leak, which way is it going to go? Out. Okay, and what would that do to our resting or to our membrane potential if the inside of the cell is losing a positively charged ion? Make it more negative, right? Absolutely. Okay, so and then the other is true as well, right? If we have a lot of sodium on the outside, not much on the inside, when it leaks, which way is it going to leak? In. It's going to come in, and that would have a positive effect on the membrane potential because we're gaining a positively charged ion on the inside. So if we have more passive leakage of potassium, meaning we're losing more of a positive charge than we are gaining, that would be a net negative effect on the cell, right? So this is like a net negative on the charge, right? And so that is one of the primary reasons that the cell stays negatively charged, is because we're losing more of a positively charged ion than we are gaining. Okay, that's number one. And then a lot of you also said C. Okay, so C, the sodium potassium pump, moves sodium and potassium, but it does it in an uneven fashion, right? It's sending 3 sodiums out and only bringing 2 potassiums in. So that's also a net negative, right? Net negative effect on membrane potential. Every time that thing pumps, that's -1 charge to the inside of the cell, right? So those 2 factors are 2 of the key factors for keeping membrane potential where it's at. Okay, and then E says the fact that sodium leaks in and potassium leaks out, and we already established that that was true as well. Okay, all right, questions on that at all? The only other thing that I didn't mention here, but I mentioned before, the third factor that keeps membrane potential negative is these negatively charged proteins that we talked about. Those are just kind of stuck in there. Those aren't moving anywhere. We have more of those negatively charged proteins on the inside. Okay, so it's really those 3 factors. It's the uneven nature of the sodium potassium pump. It's the more leakage of potassium than sodium, and it's the negatively charged ions that are stuck there. Okay, that's what keeps our membrane potentials negative. Okay, questions about that? Okay, so this is kind of a good starting place for the nervous system here. One of the things we'll talk a lot more about later is these things called action potentials. Okay, action potentials really are the language of the nervous system, right? This is when you think of a nerve signal going from one nerve to the next, or one nerve out to your muscle or out to a gland or something like that. It's always action potentials. That's what it is. This is a rapid, spreadable change in membrane potential in the positive direction. So we go from that -70, kind of showing it down here, and it rises up like this in that positive direction. And we call that depolarization. When we depolarize the membrane, we move it away from the negative direction into the positive direction. Okay, so it's depolarization. Don't get too hung up on all those ions floating around there. We'll get into the details of that a little bit later. Okay, but just for now, know that this is depolarization. Yeah, we'll get into more of those details later. Okay. Just needed to kind of introduce that because we're going to talk about action potentials here right now. So I'm going to, this is kind of a long process thing that you're going to work through. The next few slides are images of what's happening. Okay. And so what I want you to do, I'm going to show you another video on this that takes us through these steps of all the events that have to happen at the neuromuscular junction to activate a muscle cell. And then you can kind of use these steps here that's on your notes, as well as what you see in the video. And then we'll spend some time with you guys kind of filling in what's happening on these steps here. Okay, so let's go back to the laptop here and we'll watch this video.
Is also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
All right, so that's the steps there. You got to kind of see how it looks in an artistic depiction of it. So go ahead and let's work through. Sorry. Let's work through. It's surprisingly loud for that speaker. Everybody can hear it so well. But, so let's work through these next slides, kind of fill in what's happening as far as the steps from this slide. And then we'll actually have you answer this question here on the clickers at the end as well, right? So open that up, give a few minutes to send that in, as well as answering the question on the clicker.
Yeah, I know for sure it's that. I know these are stars.
This might be the chat. Or, because I don't think it's on. So maybe. Yeah. I was like 5 and 6. Yeah.
Okay, so once you've worked through those slides, then remember you're ending by answering this question here.
I think it is. So there's only steps because yeah. Yeah. That's making it more positive. It would have to be the more.
All right, so got about 30 of you so far that sent that in. Make sure you're sending your answer into this question here.
No, I think we're going.
All right, about 10 or 15 more seconds. All right, so we'll come back to that clicker question here in a sec. I'll leave it open in case you still need to send it in. But let's just briefly talk about what's going on here. Okay, so which steps did you guys say were happening here? 1, 2, and 3 as well. Okay, so we've got the action potential coming down. That's kind of depicted by these little purple lines, right? Our action potential coming down the neuron. Okay, that is going to stimulate these voltage gated calcium channels. Okay, so just like how we talked about when calcium binds to troponin, and that totally changes the shape of that protein, right? Same idea here, but instead of something binding to it, it's the electrical charge around that protein, totally changes its shape, opens it up. There's a lot of calcium outside there and calcium rushes in. Okay, and so then it's the calcium that stimulates these vesicles to move to the surface and dump their contents out, right? So what was step 3? Step 3 was calcium causes acetylcholine to release this acetylcholine into the synaptic cleft. Okay, good. So we got 1, 2, and 3 here. Okay, let's go to the next one. So now we see acetylcholine bound to those acetylcholine receptors. And we see this is a non-specific cation channel that allows both sodium and potassium to flow through. Okay, but since there's a much stronger push for sodium than there is for potassium, we get a lot more sodium going in than we do potassium going out. Okay. Any idea why there's such a stronger push for sodium? Wesley, what'd you say?
The concentration gradient, because there's so much. I mean, we're constantly pumping out 3 sodium for every 2 calcium, which just already means there's more sodium
Okay.
out than potassium, and then
So we have a stronger concentration gradient,
the
maybe.
concentration gradient is kind of allowing it to get more to.
Okay. That's one part of it. What's the other part? What else makes ions move? Their electrical gradient, right? Sodium's positively charged, the inside's negatively charged. So it's going with its electrical gradient, whereas potassium's going against its electrical gradient, right? So we have, in the case of sodium, we have both the concentration gradient and the electrical gradient pushing it in. Potassium is the opposite. We have the concentration gradient, but not the electrical gradient. So we've got a lot stronger push for sodium. Okay, so we're seeing that happening. We're seeing depolarization, right? That's what this is kind of depicting as depolarization, or moving in the less negative direction or the positive direction, however you want to say that. Okay, and that's what we see happening there. Okay, so which numbers were that? What was that? 4 and 5, is that what you guys said? 5, stimulation of acetylcholine, yeah, action potential, yeah, not quite 6 yet, but 4 and 5. Okay, all right, then we see here, once we get over to this other part of the membrane, there's different types of channels, and we'll talk about those later, but those are different types of channels, and those are the ones that create the action potential for us. Okay, so now we get our action potential. So that would be number 6, right? Yeah, so we got number 6. Okay, so we got our action potential. Okay, what else? What's number 7? Yeah, it spreads along the sarcolemma. Yeah, we see that happening here, right? Now we're back to what we've already learned about, right? Remember the signal going down those T tubules and then causing calcium release. Calcium goes to troponin. So now we're connecting back to what happened before all that stuff with excitation, contraction, coupling that we talked about. Okay, so yeah, so that was really 6 and 7, right? Yeah, and then we move on here, the very end of it. We got acetylcholine esterase breaking down acetylcholine. Okay, some of it just diffusing away like the video talked about. So this would be step, what is it, step 8, and yeah, step 8 and 9 probably here. Okay, questions on any of those? Yeah, go ahead, Ava. That's not something I would test you on, but it's choline and yeah, acetic acid were the 2 things the video said, but don't worry about that too much. Yeah, Ava.
For steps 4 and 5, for potassium, why is it going out? And
Yeah,
because I thought it's
it is. So its concentration gradient to push it out is stronger than its electrical gradient holding it in, but not as much of it moves as sodium, because eventually it's not gonna be stronger than the electrical gradient and it stops, but sodium's got both of them going for it, so a lot of sodium rushes in, right? Yeah, great question. Yeah. Okay, other questions on those? Okay, and then here, what'd you guys say? So we're kind of 47% of you saying B. We got 34% saying D, and then 19% saying A. Okay, talk to each other about that for a second. After what we just talked through and talked about, what's the primary cause of that membrane potential going more positive in this situation? Okay, change your vote if you want to. Just give you a minute to less than a minute to do this. Just change your vote if you need to.
So when the toxic pushing out, getting more negative. But.
All right, change your vote if you want to. See if you guys can. Okay, now we're up to 57% saying B, so we're a little bit better. So B is definitely the right answer here, okay? Potassium rushing out of the cell would make the cell more negative, right? Because it's losing a positively charged ion, okay? The sodium potassium pump is an active process that doesn't cause depolarization. That's trying to help us reset from being depolarized is what the sodium potassium pump does, or just maintain resting membrane potential. So not the sodium potassium pump, but definitely this here. Okay, we're going to talk about just briefly 3 pathologies related to the neuromuscular junction. First one's called myasthenia gravis. Okay, myasthenia gravis is an autoimmune disorder where our immune system destroys those acetylcholine receptors on our muscle. This leads to paralysis, weakness, droopy eyelids, muscle atrophy. It can lead to death if it attacks within our diaphragm, right? So we can't contract our diaphragm, we can't breathe, right? So this is a autoimmune disorder. Tetanus, this bacterial infection that where most of us are vaccinated for, it releases a chemical that binds to acetylcholine receptors just like acetylcholine does, but it doesn't let go of it. It just holds on to those receptors, causes our muscle to contract in a very forceful, violent manner, so much so that it can break our own bones, right? It can actually rip our tendons right off of our bones, cause our bones to break. This is very, very forceful contraction. Again, if that toxin gets into our diaphragm, then it can be fatal, right? Because that diaphragm just contracts, you can't breathe, right? And then lastly, Curair, which is a acetylcholine receptor antagonist, which means it gets in the way of acetylcholine binding, but it doesn't do the same thing. It just blocks it. Okay. This is found in those little poison arrow or poison dart frogs in South America, right? It's on their skin. You touch them, you die, right? Because it gets into your body and you can't contract muscle anymore. Your heart can't contract, your diaphragm can't contract. So it's also one of the ingredients in lethal injection. Right? When they have the death penalty carried out on them, one of the ingredients is this chemical because it makes it so your diaphragm can't contract anymore. Right? Okay. All right, guys, we made it. I'll leave that up so you can finish writing it down if you haven't yet. Good luck in lab if you haven't been there yet, and we'll see you guys on Friday. All right, see you, Talia. Bye.